warm open this time?
I think warm. I think warm
Cozy?
is good.
Cozy, warm open?
Sweater weather.
Welcome to the Saber Podcast. My name is Lily.
And I'm Ren.
And today we're talking to Dr. Bunnick. Dr. Bunnick is an associated professor at UT Health San Antonio. She earned her PhD at the University of Amsterdam, where she studied neutralizing antibody responses in HIV-infected individuals. Since then, Dr. Bunnick has researched various topics in immunology, parasitology, and functional genomics, and she has developed a resource program dedicated to unraveling the nature and acquisition of protective B-cell responses in malaria-experienced individuals.
Yeah, Dr. Bunnick is really cool. She taught us some of our immunology lectures back in the first semester, and that was a ride. So we hope you enjoy this conversation.
Absolutely. This is, I think, amongst our episodes, a little bit more like microimmunology, basic science based.
Definitely, yeah.
So it's just fun to hear about her research, I think. For sure.
So we'll start our conversation with her just kind of letting her talk about her journey in research and what she works on and how she got there.
Yeah, well, so I started working on HIV because I was just really interested in the virus, and I was interested in how people sort of respond to the virus, what kind of immune responses people make once they're infected. And so that was my PhD research. And then at the end of my PhD, I had sort of the choice to either continue in that field and just dive deeper into HIV, antibodies, or HIV virology. But really, I wanted to learn a lot of new things. And so I decided to switch fields. And I decided to, instead of study HIV, to study malaria. And sort of the reason I ended up with malaria is because I am interested sort of fundamentally in diseases that impact a lot of people in the world, and especially people living in resource-poor countries. And so that made sense for HIV, but it also made sense to then switch to malaria. So that's kind of how those two are mostly connected. But in terms of diseases, they're very different diseases. And initially, I didn't study immune responses in against malaria or against malaria parasites. When I chose a lab to do my postdoc, I was mostly interested by the technologies that lab used. The lab used a lot of next generation sequencing approaches to study basic questions of the malaria parasite, how it survives, how it grows, how it replicates, how it controls gene expression. And that's what I really wanted to learn. So that's how I rolled into the malaria field, but learning totally new things that have benefited me in my current research. So I learned how to do next generation sequencing and how to do data analysis. And then when I was at another crossroad where I was deciding what I want to do as an independent researcher, I kind of combined my expertise from looking at antibody responses in HIV infected individuals with the knowledge that I had acquired in the malaria field and the tools that I had learned. And that's how I ended up studying antibody responses in malaria exposed people.
That's really cool. Mm-hmm. What a winding road. They are indeed so different. The projects we were looking through, HIV is very different from malaria, of course. The antibody responses, could you speak a little about sort of, I don't know, similarities, differences? What do you find most fascinating about the responses that you study?
Yeah, well, so the main difference is that HIV is a true chronic infection, whereas people can be treated if people have malaria or people have Plasmodium infections, they can be treated, clear the parasite, or even if they live in malaria endemic regions and have sort of lifelong exposure, they develop really strong immune responses and they're able to clear the parasite by themselves. And so that's a clear difference. But in terms of how the immune response develops and how the immune system responds, it's actually very similar. So we see similarities in B-cell responses between people who have HIV infections that are chronic versus people who have sort of repetitive or semi-chronic Plasmodium infections. And it has kind of the same impact on the immune system. And it's one of the things that we're really interested in seeing. So my lab studies, on the one side, how B-cell responses and antibody responses protect against, mostly against disease, not so much against infection. And how those antibodies work, like where do they bind on the antigen and exactly how does that lead to what are the molecular interactions and how does that lead to parasite inhibition. But on the other hand, what's the effect of having this parasite in your body chronically or for long periods of time, repetitively, and how does that change the B cell response? And how does that sort of continuous inflammation change the B cell response? So it's kind of both sides of the coin that my lab is interested in.
So Dr. Bunnick actually came out with a paper May of last year, 2024, that investigates B cell responses to malaria. It's one of the papers that we found particularly interesting.
Yeah, it's a pretty big one too in Nature. So.
Yeah, exactly. So we just wanted to know the story behind it, and it's a pretty cool story.
I was at a conference and I was presenting. I had a poster about sort of some of the tools that we had developed in the lab that we use to identify antigen-specific B cells, and then we can sort them and clone, isolate the monoclonal antibodies from those B cells. And we had applied that to a totally different antigen or a set of antigens, and I was presenting those data. And then someone came up to me and said, "Well, this is all really cool, but can you do it for this one protein that I've been working on for all my life?" And I was like, "Well, I don't know. We can try." So he sent me some protein, and at the time I had a graduate student who had just started. He was in his first year, and I said, "I've got a project for you." And so he started isolating monoclonal antibodies for this particular antigen, and it turned out that he found some really, really cool antibodies that have very broad reactivity, because this one protein that we're looking at, it's not just one protein, it's a whole family of protein. There's a lot of variation in sequence, and it's a really good vaccine target. But the problem is that there's so much variation that if you immunize a person with one variant, that it makes good antibodies against that one variant but nothing against all of the other variants. And so the goal is to, can we somehow figure out how to make antibodies that are broadly reactive? And our starting point was by just isolating those kinds of antibodies from people who have had malaria, and then kind of working back to design the immunogen that can elicit those kind of antibodies. So the process that we're inhibiting with these antibodies is the adherence of infected erythrocytes to the endothelium. Okay. And so this is what underlies the most severe complications that you see in malaria patients. So the parasite, as it infects erythrocyte, it expresses a lot of proteins, and some of those proteins end up on the surface of the erythrocyte. And when the parasite adheres to the endothelium, and it does that in large numbers, it basically blocks the capillaries, and it blocks oxygenation of the tissues. And it also elicits a response from the endothelium, so you get a lot of inflammation, the release of cytokines and clotting factors, and it's a mess. It's really, there's a lot of bad things happening that ultimately lead to people dying as a result of these processes. And so this process is the parasite that we study, Plasmodium falciparum, it's human specific, and so you cannot just put this parasite in a mouse model or in another animal, because it doesn't do the same thing. It's not able to replicate, and it doesn't have the same attachment to the endothelium. And so the collaborators that we're working with in Spain, they basically make human capillary, they make a microvasculature in collagen. So they have they have a little network, a microfluidic network in collagen that they can seed with human primary endothelial cells that basically form little capillaries. And then if you perfuse that, your infected erythrocytes will bind to the endothelium, and they can measure that. And then if you perfuse them with our antibody, you can measure inhibition. So you can see how now the parasite is not able to attach to the endothelium.
That's so really fascinating. Cool. That's so cool.
Because you can show the attachment and inhibition on a molecular level, but now you're able to prove that it would work kind of organ system. And
exactly.
Yes. That's awesome.
Yeah.
Very cool.
And then just to sort of come back to your initial question about the Tanzanian serum samples. So we, as we had submitted this paper to Nature, or the manuscript to Nature, one of the reviewers asked us for sort of an estimate of how prevalent are these kinds of antibodies in serum from people who live in malaria endemic regions. And so this is where the Tanzania serum samples come in, because we try to address that question by using samples from people living in Tanzania who have malaria. or who've had malaria, and just trying to get a sense of sort of when these antibodies develop and how prevalent they are.
It just seems
Super
like a
cool.
really cool project, and
Mhm.
like a lot of global involvement too.
Yeah, absolutely. So we, a lot of our samples, or most of the samples that we work with here, are from people living in Uganda. So we have collaborations with our main collaborators on that side are actually at UCSF and Stanford, but they have cohorts in Uganda where they basically continually monitor people, and every month or every 3 months, depending on the setup of the cohort, these people come to the clinic and they donate blood, and the material is stored. And so we have access to all kinds of samples from children, from adults, sometimes longitudinally, that we can use for our research.
Wow.
So yeah.
That's interesting. How does this is kind of derailing from the research itself, I suppose, but the nitty gritty is interesting as well. How do those relationships form between these like groups where you go into Uganda and like set up camp essentially to draw those samples? How does that like logistically work out?
So I'm not entirely sure how they have set this up, but this cohort has been running for a long time. And I think they started probably around 2005, like 20 years ago, they started setting up these cohorts, and they were already well up and running when I initially approached them. So I spoke with, when I was setting up my own lab, I had discussed with my then postdoc advisor about, if I'm going to do this, I'm going to need access to samples. And so she used sort of her connections in the field to introduce me to people who she thought might be willing to share samples. And fortunately, the people at UCSF have been super generous. Like, they've never said no when I asked them, like, hey, this is the study we would like to do. Can you help me find the samples that would fit those criteria? And then they've gone through their databases, and yeah, they've always been able to provide us with the samples that we need. The antibodies that we've isolated, we've actually been able to share them with them because they were interested in using them for their research. So I was like, oh, I can finally do something back for you.
Yeah, that's
Awesome.
really cool.
Really nice.
So that was the Nature paper, and it was really interesting stuff, cool findings. We wanted to know kind of where her research is headed next, what the next steps and final sort of endpoint of her work is going to be.
Yeah, well, ultimately, what we want to do is develop a vaccine.
Right.
So, and this would be a vaccine that prevents severe malaria, so prevents the most severe complications. So it basically prevents young kids from dying. That's the idea. We are, with the antibodies that we have and with the knowledge that we've gained from looking at their structures, we have started to design immunogens. And when I say we, it's in collaboration because we are not an immunogen design group, but we have sort of as a consortium, we're now collaborating with people who have the tools and the know-how to design these kind of immunogens. And specifically, we're collaborating with Neil King at the University of Washington, who has this really cool artificial intelligence driven protein design pipeline where he can take the structure. So he has our structure of how our antibodies interact with the antigen. And based on that, he can sort of model and design like minimal epitopes or really small proteins that have the right conformation. And so, this pipeline initially spits out, I don't know, tens of thousands of options, and then it sort of selects down, and then like a manageable number can be produced recombinantly and then tested whether they bind to our antibodies. And so we have a few hits now that we're hoping to test further, see if they're immunogenic and if they elicit the right kind of antibodies. So that's going on in the background, and that's sort of out of my hands, right? It's other people who are looking into this who really know what they're doing. But for my lab, we are still continuing to isolate more antibodies because so the antibodies that we isolated, they're really broad and we're really excited, but they're not as broad as we would ultimately like. Like, there's one group of proteins that does not bind these antibodies. And so we want to find better antibodies. So we want to continue search for antibodies that truly bind to all of the potential variants within this protein family. We're also interested in how these antibodies develop, because the antibodies that we have isolated were from adults who have 20, 30 years of Plasmodium exposure. And so over time, there's a lot of B cell maturation, a lot of changes can happen in your antibodies, a lot of mutations get introduced. And so we're interested in seeing, especially in young children, the very first time they get infected with a Plasmodium parasite, what's the response then? And then how does it develop over the next couple infections? And so with our collaborators in Stanford, we're hoping to address this question. So they have a birth cohort where they follow children from the moment they're born up until they're 3 years of age, and they're sampled every month, and they know exactly when they were infected. And so what we're going to do is we're going to look at sort of 3-year time points, see which kids have good antibody responses, and then kind of go back in time and see if we can track how those antibodies developed. Wow. Which is something I'm really excited about.
That is really exciting.
Super interesting. So the reason I wanted to talk to Dr. Bunnick is actually because I came across an article about her lab during the COVID-19 pandemic, where her resources and her antibody library was kind of co-opted into a study about COVID-19 and about how the virus sort of generates different immune responses in different patients, people who get very severe disease versus mild. And it's a really interesting bit of work that kind of predates and coincides with the Nature paper we just told you about.
Yeah, so when the pandemic started, we thought it would be really interesting to use the tools and the methods that we had developed for our malaria research to see if we could apply those to, look at immune responses in people who have COVID. And so we collaborated with Tom Patterson, who is the chief of infectious diseases, and basically set up sort of a little cohort study here where we got samples from the patients that were in his clinical trials. And we collected over, I had a graduate student who was doing most of this work, but he sort of single-handedly by himself collected samples from more than 100 people, and a lot of those people actually came to we had a sample from the moment they were released from the hospital, and then they came back 2 weeks later, or some of them even came back 2 months later. So we had some longitudinal samples as well, which was interesting. But yeah, it was a lot of work.
Absolutely.
But at that time, we couldn't really work, or we were not supposed to work on malaria, right? Because a lot of the lab activities were shut down, and we had to sort of figure out logistically, we couldn't have that many people in the lab. So we had to sort of make a schedule. Had a few people working from home at the time, and then he was in the lab almost full time working with these samples. But now that we had that large collection of samples, we wanted to look at the B cell responses, and again, using some of the tools that we had developed for our malaria research, and see if we could sort of what we could learn from how these B cell responses developed in people who had essentially their first infection, right? It's a new pathogen. No one has any pre-existing exposure. So what does that look like, and what kind of B cells, what kind of memory B cells form after this kind of infection? And the main question that we asked was, what's different about people who have a relatively mild infection? So all of these people were hospitalized, so they were all, we called them non-severe, because we didn't call them mild because they were still hospitalized. So we didn't want to say they had mild infections or mild disease. So we called them non-severe versus severe, which are people who were on ventilators and who needed assisted breathing. And the idea was that, okay, so these are people who have the severe, people with severe disease really have a ton of inflammation, kind of what you see in patients with severe malaria, versus the people who have, maybe they're sick and they have, there's a lot of things happening, but they're not quite as, they don't have the same level of inflammation as this. Anyway, that was the main question, like what, and I guess the main application as well to other diseases is how do B cell responses develop in the context of high inflammation versus lower levels of inflammation.
Yeah.
And I think that would be the main, I guess, thing that we have learned from this is that you definitely see differences in terms of how B cell memory develops depending on how sick you are. Mm-hmm. So we don't think that, that was the main message that we got from that study.
And then this is the part of the conversation where she gets more into the nitty gritty, the details about different memory B cell responses that are still being kind of investigated today.
It's mostly so within the B cell field. It's sort of increasingly being recognized that there's not just one memory B cell, right? If you look at a sort of an immunology textbook, there's a naive B cell that becomes activated, goes to a germinal center, it can make a decision whether it becomes an antibody secreting cell or a memory B cell, and kind of that's it. But we've now started to appreciate that there are different flavors of memory B cells. Like some memory B cells are more prone to rapidly develop into an antibody secreting cell. Some memory B cells are very long lived, other memory B cells are a little bit shorter lived. And we're trying to identify those different flavors of memory B cells by looking at which surface markers they express. And one of the surface markers that had already by others been associated with long lived memory is called FCRL5. And that's the marker that we saw increasingly expressed on memory B cells in people who had non-severe disease. So they make better memory responses, more long-lived memory responses than the people who had severe disease. And so having severe disease and having that really those high levels of inflammation probably interferes with your ability to form good memory B cells.
That makes sense.
And so in the context of like immunization development, I'm guessing that there's an interest now that there's an understanding of the different flavors of memory B cells in specifically kind of activating pathways that go towards the long-lasting memory B.
Yeah, no, it's definitely something that my lab is very interested in because especially in the malaria field, but this is true for other vaccine fields as well, we have vaccines that work well for a very short amount of time, right? They elicit strong immune responses, but then those immune responses wane very, very quickly. And so how do we make long-lived memory B cell responses and long-lived plasma cell responses? Those are things that we don't understand very well, and we would like to get a better handle on.
Yeah.
So after asking about her work with these B cells and coming to understand kind of what she's doing and what she's looking for, we were curious what it would look like when the day comes that, the vaccine is found or the right antibody is identified. What does actually, getting the product out there look like?
And the really cool thing about malaria, as she'll go on to tell you, are the models and the ways that we can test how the vaccines work in real life.
So once we find that, we would have to make the immunogen that can elicit those kind of responses and then test it in the field. And initially test. The nice thing about malaria vaccine development is that we have what's called controlled human malaria infection models, which is basically you immunize people and then you infect them with the parasite. And the nice thing is you can treat them, right? There's treatment as long as you infect people with a drug-sensitive parasite. You can infect them for a short amount of time and you monitor them very closely. You don't let them develop full-blown malaria. You can just, there's PCR-based methods that are very sensitive, so you can detect parasites in the bloodstream before they even know that they're infected. And so we can test vaccines and vaccine efficacy in those kind of trials. We can test safety and immunogenicity and even some efficacy in those very controlled settings. And then if the vaccine, like the candidate vaccine, kind of passes that hurdle, then we can take it to the field and see if it works in a real life situation.
Exciting.
That's exciting.
And when we asked Dr. Bunnick what students should do if they find this kind of research particularly interesting, she said,
well, I'd say just do it because it's super cool.
And that's that. So it was a pleasure talking with Dr. Bunnick. We hope you enjoyed as well. We're going to include some links to the Nature article and the COVID paper that we read,
as well as some kind of basic immunology stuff, because I know that I'm not an immunologist and it's been a minute since I took the class.
Lots of terms, lots of words. If you're not familiar, these resources will definitely get you a better idea of what Dr. Bunnick's been talking about.
For sure.
So cheers to that. And thank you for joining us on this episode of the Saber Podcast. We'd love to have you back again next week. So be sure to subscribe and leave us a review if you feel so inclined. If you have any questions, email us at saber.podcast.outreach@gmail.com or find us on Instagram @saberpodcast. Until next time, I've been Ren.
And I've been Lily.
And stay tuned for more inspiring stories.